The overall goal of this procedure is to induce chondrogenic differentiation of ASCs for defected cartilage repair.
This method is to induce the pre-chondrogenic differentiation of ASCs using centrifugal gravity force. Though this method can provide insight into chondrogenic differentiation of ASCs, it can also be applied to other systems such as osteogenic and fibrogenic differentiation.
The main advantage of this technique is that chondrogenic differentiation can be easily induced from stem cells without treating any cytokines.
Demonstrating the procedure will be Yeonsue Alice Rim and Yoojun Nam from my laboratory.
Begin by harvesting 80% confluent adipose-derived stem cells from a 100 mm culture dish by discarding the medium and washing with 5 ml of PBS. Then add 1 ml of PBS containing 1 millimolar EDTA and incubate for 2 minutes at 37 degrees Celsius in a humidified incubator containing 5% carbon dioxide. After 2 minutes, tap the culture plate gently, then add 4 ml of fresh medium and transfer the cells to a 15 ml conical tube. Centrifuge the cells at 250 times G for 2 minutes at room temperature. After centrifugation, remove the supernatant without disturbing the pellet, then resuspend the pellet in 10 ml of medium. Count the cells using a hemocytometer.